Good morning, everybody. I'm Deepthi Rajagopalan. I'm a bioinformatics specialist at Dave Lab in Duke University. Today, I'm going to talk about defining the genetic overlap of patient tumors and patient-derived xenograft models in colorectal cancer. So colorectal cancer is one of the most common forms of cancer. Studying the molecular basis and genetic heterogeneity of this cancer will help us understand the mutations that drive this cancer, as well as give us insights into treatment approaches. Patient-derived xenograft models are created by grafting the patient tumor on an immunocompromised mouse. PDXs are an important tool to study tumor growth, progression, and response to therapy. So our aim was to evaluate PDX as a tool to study cancer. So we wanted to compare tumors with the paired normals and PDXs. We wanted to identify recurrently mutated genes previously found in colon cancer. We also wanted to identify copy number alterations and deletions in tumor normal and PDX normal pairs. So when we think about tumor samples in general, we tend to think about them as a homogeneous entity. But in reality, they can contain normal cells as well. So here, the red cells are the tumor cells, and you can see that there are some normal cells with them as well. Similarly, when we think about PDXs, along with the tumor cells, there might be some mouse normal cells. So for this project, we started out with Illumina HiSeq, 125 base paired exomes. We had 7 tumors with their paired normals and PDXs. So in the first step, we mapped simultaneously the samples to mouse and human genome. Anything that mapped to the mouse genome were discarded, and we looked for somatic variants in those that were specific just to humans, and also looked for copy number alterations. So speaking about the quality of our data, from the graph on the left, you can see that on an average, we got about 50 million reads. And from the graph on the right, you can see that our average targeted depth of coverage was 50x. Then we wanted to compare the somatic variants found between tumors and PDXs. So this plot shows you the counts of the different types of variants that were found. So this is the count of non-synonymous mutations, synonymous variants, and some stop gains and unknowns. So we also wanted to cluster these mutations. So when we did a hierarchical clustering at gene level, we saw that the tumor profile was more similar to its corresponding PDX. Then we also wanted to look for some known cancer genes. So we specifically looked for cosmic mutations in our data set. We were able to find 42 such mutations, and we saw that majority of the ones that we found were found both in tumor and PDXs. Then we wanted to look for hotspot mutations that were specific only to colon cancer. So we found KRAS, NRAS, PIK3CA, shared all between tumor and PDXs. But in case of P10 and TP53, they were found only in the PDX and not in the tumor. Similarly, SMARCA2 was found only in the tumor and not in PDX. So when I looked further into what was going on, I saw that these variants were present, but they were just found in really low allele frequency. So when we looked at copy number variation, we found that significant amplifications and deletions were highly similar between tumor and PDXs. To give you an example, here is chromosome 20. So we see an amplification in the Q arm in the tumor. We were able to identify similar amplification in the PDXs as well. So in conclusion, we found that genetic mutations and copy number alterations from the tumors were preserved in PDXs. However, coverage and tumor purity can be a limitation in such studies, and tumor-specific PDXS model do recapitulate the heterogeneity of a primary tumor and are suitable models for development of targeted therapies. Thank you so much for listening to my talk. I'd like to acknowledge the members of Dave Lab. We have a couple of posters, so please come visit us. We're also looking for more people to join our team, so if you're interested, definitely visit our booth. Thank you.